All right, so we will move on to our next section. So with that, and Brian's already ready, I would like to introduce Brian and Elizabeth from the Los Alamos National Laboratory team, and they will present on the HIV Sequence and Immunology databases. So Brian, take it away.
Okay, let me get my screen sharing started here. Share. Full screen. All right, this is about the HIV sequence and immunology databases, and we'll mostly focus on the immunology database because that's what most of the audience here is interested in. But the two databases are quite integrated together, so we'll talk a little bit about that.
Sorry, Brian, the wrong screen sharing.
It's not sharing well?
We're seeing the back end of your presentation rather than the front end.
Huh.
You see the presenter view.
Okay. Presenter view. Where do I go for that?
I think stop your screen share and just redo it and click
okay.
The other screen.
Share screen. Is that right or not?
Yep, that's perfect. Thank you.
Okay. And I'll go back one slide. All right. Anyway, the databases have about 9 employees. Most of us are PhD scientists. We're mostly virologists or sequence analysis people. So we're very much biologists and not computer scientists or not programmers. We have 1 or 2 programmers and scripters on our staff, but the focus of our employees are mostly biology. So you can write to us with biological questions if you have them, even experimental design and so on. Our databases are updated from the literature. The sequence database, we download sequences from GenBank, and the immunology database, we scan the literature as the IEDB does. And we're specifically looking for anything related to immunology, immunodeficiency viruses, and epitopes, antibodies, CTL, CD4 epitopes. And we enter data from the publications into our database. I'm not able to control my slides now. Okay, just clicking does it, sorry. I can't go backwards, so I guess I can't, here I go. All right, there we go.
The cursor still work, okay, yeah.
Yeah, here we go. Sorry about that. So we have antibody epitopes from thousands of papers. We have CTL epitopes from hundreds of or thousands of papers also. And we have almost 1 million sequences of virus sequences now in the database. And what we're seeing on the right of this slide is the virus sequence down there at the bottom. MGARA is the GAG gene, and the towers above that are all the epitopes that have been defined to bind to various peptides, typically 9-mers in the virus. So the sequence database has almost 1 million sequences now, and there are various ways to search that, and I'll talk about that later after Elizabeth does the immunology database introduction. The database was founded in 1986, the sequence database. When Jerry Myers realized that HIV was going to have a lot of diversity, he decided that GenBank wouldn't be enough, that we would need a separate database for viruses, for HIV, and he started that early on, which was a great help because he really helped with the nomenclature and taxonomy of the viruses too, to keep everything nicely organized. I pity people who study some other viruses. You go to GenBank and you download thousands of sequences, but it's hard to get like a subtype reference set or help with the taxonomy and classification. It took me about 3 weeks to figure out what SARS-2 was like, where it fit into the overall picture of coronaviruses when it first came out in January. If we had a subtype reference set of all coronaviruses, it would have been a great help. And then the immunology database was founded in 1994 by Betty Korber, and she was an immunologist and a statistician, and she's been a huge driver of our immunology database. And this database pair, the sequences of the viruses and the human immune response, and somewhat with macaque responses in experimental models, is a unique resource because we have thousands of patients that have been followed over time. We have populations of people. We have molecular clones that have been injected into macaques and so on. So we've attacked the global research community of HIV has attacked the problem of host virus response in numerous ways for dozens of years now. And there's really a rich body of data to deal with there. Beyond HIV, our tools, most of them, or a good portion of them, are applicable to other organisms as well as HIV. So we have many tools that can be used for any virus, any pathogen, and our phylogenetics tools are good for any organism whatsoever, any DNA sequences. And we also had a hepatitis C virus database, and that is still active but no longer funded. But you can view that and interact with it here at our website. We have a hepatitis C subtype reference set and so on. We have also a database of hemorrhagic fever viruses, filoviruses, and so on. So you can come to us for help with many other viruses. Also, if we don't have a virus listed, but you're interested in it, like dengue or something else, you can come and ask us questions, and it's quite likely that we'll have some ability to help you with those. Now I'm going to turn it over to Elizabeth, and she'll have to start sharing her screen, and she'll tell you about the immunology database.
Okay, hello, thank you very much to IEDB for giving us this opportunity once again. You've invited us year after year to share our database and to show you mostly immunology, but also a little bit of the sequence. And as Brian said, there's the immunology portion and then there's the sequence portion of our database, and they both are very linked, and that actually gives it a lot of power. But today we're going to focus a little bit more on the immunology, which is where the epitope information as well as some other things are reside. That information resides there, but the two crosstalk so easily and so seamlessly that very often as a user you won't even know that you're going from immunology to sequence and back, and it really doesn't matter as long as you get what you need. So I wanted first to show you what we'll be talking about. Brian gave you an overview, and then I would like to go on to talk about what we have in the immunology database. We have something about T-cell reactive epitopes and some tools that are there to deal with them. And then we have some information on antibodies and some tools and other resources for neutralizing antibodies. And then Brian will come in later and just give a very brief bit about the sequence database, and then we'll be open for questions. So if you were to go to our database, which is at hiv.lanl.gov, this is the page that you would see. I wanted first to say, if you have any questions, please don't hesitate to email us at immuno@lanl.gov. And Brian will be sharing later on the sequence database questions, and you can ask it questions from any of us. We'll get the answers to you. So if you go to hiv.lanl.gov, you see this page, and it is from here that you can go into the sequence database, the immunology database, or other viruses. There's usually the latest in news at the bottom. And if you were interested in immunology database, to click here. But I'm first going to take us on a little diversion just for a couple things about how we curate this database, so that it's easier for you to use later on. And then we'll get on into how to search this database. So, and I'm going to be really quick and fast because we have a lot to cover in a very short time. And so I just want to touch on a few things, but please feel free to ask us and email us if you need. So this next slide just tells us how the database entries are curated and annotated. If you were to look at T-cell epitopes and their annotation, you would see that every epitope HLA combination has a record number, a different record number based on each study. So each paper or publication or study, along with the epitope and its restricting HLA, form one record number. So it is possible for one epitope to have really many, many record numbers. And this is different from how the antibody information is annotated. For each antibody, no matter how many studies there have been on it or how many studies have been annotated, there is just one record number. And this is because biologically speaking, a monoclonal antibody is a monoclonal antibody, no matter which study, it's the same molecule. But as far as the T-cell reactive epitopes, there are so many different contexts, which patient, whether there are variants, whether it's a different HLA, just so many different contexts. And so each study is a different record, but not to fear that these different epitopes and their record, even though they have different records, you can find all the study information very easily. Also in our database, we have many different ways of searching it. And we also have information on sequences. We have information on variants, which could be escapes or not even escapes. We have information on the HLA and the MHC, and then a lot of antibody resources. And I know I touched on these earlier. There are five different search interfaces through which you can search the epitope variants or the antibodies, or even whether they're CTL or helper epitopes. And then there are many, many tools and maps where you can see where the epitope lies on a certain protein, HIV protein. Of course, that's HIV specific. And then there are these other neutralizing antibody resources. And in this next slide, I just want to give you our latest news. And that is that there's a new tool coming up where you will be able to programmatically retrieve any of our curated epitope data along with variant data. And so whether it's antibody epitope or T-cell epitope, you should be able to get this. This will make things a lot easier especially for those of you who are interested with multiple searches and automating your searches, because you'll be able to get this information and then do your search. And it should also help with us being able to interact with other databases and mix and share information. So we're looking forward to that. It's in the form of JSON and it's an API interface, and that should be live pretty soon. It's already all completed. We're just waiting for lab security clearances and things to come through, and then it's going to be out. So we're looking forward to that a lot. And then there's a few other things in the pipeline too. This was created by Jim Singer. If you want to contact him, that's his email address, otherwise immuno@lang.gov. And then on the next slide, I want to show you that we do have a lot of immunology tools. We also have a lot of sequence tools, which I have not on this slide. But what I wanted to show you was that all the tools that are indicated in purple or blue are actually general use tools. So these tools can be used for any species. It does not have to be HIV. The red ones are HIV specific. You can see that those are actually in the minority. And so this is a good site to come to, even if you're not studying only HIV. We also have links to external tools, and these links are actually very, very useful because you can actually go out and interact with other databases. Some of them are epitope prediction tools. There are germline antibody resources, immunology, vaccine study tools, many things that you can go to from here. But let's go back to our immunology page, our database page, hiv.lang.gov. If you were to click on immunology database, this is where it would take you. A series of lists for search interfaces, database products, and you could get any of these from the drop-down menu up here too. So just whichever way you like, there are multiple ways to get to this. If you were to do a search, you would get a result that would have notes and links to other information. But we'll get there later in this talk. First, let's look at the database products. There are maps, there are also tables, there are alignments, and then you can get to various mutations and things, and also some other tools. At the bottom here, I wanted to show you that we have links to some other groups' work. Sometimes they have really a lot of data. And instead of us annotating everything, if the other group is willing, we have just presented it completely in a tabulated form. So you can go straight to that from down here. But if we were to look here, you could see that there's tables and maps. And I want to show you this page again to show you other things that you can get to on this page. And for example, if you were to click on tables here, you could get an option of several different tables. And some of these are CTL tables, there are helper tables, antibody tables, and I'll show you a little bit more about the CTL tables. And of course, you could get to alignments and such too. So let's take a look at the CTL tables. We have 2 CTL tables. The nice thing about the tables is that there are the whole sequences of each epitope given here. If you were to look at maps, you would not actually see the sequence, you would just see the location of the epitope on the map. But you could click on that because they're interactive and then go to the sequence. So that's not really a problem. But here is a table. And we have 2 tables for CTL reactive epitopes. We have the B list, which is essentially everything that's been annotated. And it comes with information about it, restricting HLA, protein, locations, species, things like that. And then we have the A list. And that is curated by Christian Brander and his colleagues. And this gets curated and it is updated almost annually every time we release the compendium. We have an online compendium that is released annually. And with it is this review on the A list, which is the gold standard of epitopes. And so you can be very sure when you look at the epitopes here that they have been doubly checked and that they are well-known, annotated, and there are actually some particular attributes that they look for before it makes this list. So on to the next thing, which is the maps. This is what a map would look like. Now, this is just a little screenshot of a little bit of a map. And you can see that epitopes, which are bounded by these double arrows, reside upon this map. They show it, and it's also got the restricting allele for HLA on top. Some of them overlap, some of them don't, but each one is only mentioned once, not by how many times the study is done, but just each epitope is mentioned once. So that's another way that you can look at your epitope. You can click on it and then go to notes. And then finally, I wanted to show how you could actually do a search, like a direct search on your epitope of interest. If you were to look at this particular slide, you can see, let's pretend we want to do a search. And by the way, all these searches, you can have a fuzzy match, especially on location, so that you can find variants, you can find embedded epitopes, you can find overlaps. But let's pretend we want to do the search on our famous SL9 epitope. And if you were to put SL9 in here, you would get, and then search, you would get something like, this is an older slide, so 294 studies that were done on it and 294 sets of notes and things on it. But now if you wanted to narrow that search, you could add in, say, suppose you chose to add in a certain keyword. Then if you added in the keyword escape, so SL9 plus escape, you just get 35 entries. And so that's really a lot more manageable for a user. And so using these different choices, you can actually get a nice tight result for what you're looking for. Another thing, and that is given here in this example is, for example, here is a search for IW9, which is a pretty famous epitope. You put it in there, and then if you knew which author you wanted to, which author's work you wanted to look at. So here's Pillay. And if you put in Pillay as the first author, your result would be this. And this is a record, which gives you, and this is the canonical result, it gives you the record number, it gives you the location, and also what the author coded as the location, gives you many bits of information like subtype, what the immunogen was it in humans or not. And if we know the patient that this study was done in, if we know all the epitope, that patient's HLA, then we have that in here. And then the experimental methods that were used in that study, keywords. These keywords are these are put by us. These are not just what the paper in question has as keywords. After reading the paper, we put in these keywords so that when you, as the user, want to make a search, you can make a very tight, crisp search. And then there are notes. Here for the T cells, the notes usually are, the first note is T cell epitopes. The first note is a very short summary of the whole paper and results. And then the second note onwards, you have little short summaries of different experimental results within the paper. So that's gives you a really nice way of looking at what this study was about with respect to this epitope. Now, I'd like you to turn your attention to certain links that are present on the right side. There's one to the epitope map. There's another to epitope alignment. And then there's, if there are variants annotated, then you can look at that. We won't look at the map because we already looked at an example of the map, but let's look at alignments and then variants. So here's a result of an epitope alignment. You can see that your epitope is marked out on the HIV genome. And then what you find is you find a whole a result of the alignment of your epitope to the repository of sequences that are in the sequence database. So here's an example of where you would cross over into the sequence database without necessarily knowing you were. And you can look at your epitope related to that. And then in case you were interested in getting a result like this and aligning to the HIV sequences that we have, but you didn't want to go in through a search, you could go to the tool that we call Quick Align here on the right, and you could get a similar result. There are also more usable, more flexible tools like the Analyze Align tool, where you could put in your alignments of sequences and then look for a certain epitope, and you could find positions, frequencies, variant positions, and you could even create a web logo. And the nice thing about Analyze Align as a tool, which is a separate tool, and you could get to from the tool drop-down menu or the products list. The nice thing about Analyze Align is that you can even get discontinuous positions in your epitopes. And so this is one of the things you can see, but let's look at it. If you were to click on that right side of the results page if you were to click on variant details, let's see what you would see. You would see something like this for the study by Pille with IW9. They had reported 2 variants, I guess 3 variants, but we're showing you 2. One of them was actually an escape, which the paper said was an escape. You can see what the mutation was. Sorry, I went up a little too fast. And you can also have a very little note about what that variant did in the paper. And then here's a separate variant that they found, which was not an escape, it just had a diminished response. And these we call mutation types. And it is something that we actually codify. So we look at what it was in the paper and we see whether there was an escape or whether it was an inferred escape or a diminished response. We have many different mutation types that we pull from so that you as a user can actually search on them if you wanted to. Next, I would like to go on to antibodies. Now, I'm not going to show you an antibody search page because it's very much like the CTL and the helper search pages, which are essentially the same. But it does say antibody search on the top. So if you go to the antibody search and you searched for your own antibody, this is the kind of result that you might get. And here in this example, we're looking for the antibody CH235.9. And this was the result page of it. If you would look at it, you could see that you find the name of the antibody, location. If there is an epitope known, it would be given. Some neutralization data and details. Contacts and features, and then the patient within which it was studied, which we call the donor or the patient, details on that. I would like you to look to the right here and see that if there is an epitope alignment, which there's not an epitope here, so there's not an epitope alignment, then you could click on it and get to that. But you could always look at the epitope match because they know where this, what class of binding this antibody has, and you could look at the ONV, pretty much all of them are there, and you could look at the map and see where that many other antibodies that the epitopes are known for, where they bind. So that's available, but otherwise there are notes as well. Now keep in mind for the antibody, each note is a separate publication or study. And so you can see that here in the notes, Bonsanuri, Lamb, another Bonsanuri. And so those are notes that you can click on and you can go on to see what the notes were about that. But now let's go back here to the things that we can get to about this antibody. I'm going to go backwards. First, I'd like to take you in to see something about the donor details. We have a whole patient database, links to all our antibody and T-cell reactive epitopes that do have patients mentioned. And then antibody features database where you can link to. This shows you antibody contact positions and also tells you something that as certain protein features and I'll show you what we mean by that. And then there's a link to CATNAP. CATNAP is a tool, and within that tool resides the neutralization data, or it accesses the neutralization data that we've annotated for each antibody when we have it. So let's go on to look at patient details, then we look at antibody features, and then we'll go on to see neutralization details. So here are patient details. If you were to click on that link, you would find something like this. You could find out the patient code. This is the very famous CH505 patient. You could see who he was, which year the infection was noted, what the HLA type for the patient is, the ethnicity, the country that the study was performed in. You can even see a note on that particular patient. So that's interesting because sometimes there are numbers, ELISpot result numbers and things like that. And then you can see whether that patient is linked to antibody records, which you can see that this patient had a lot of antibodies isolated from them. And also there are some CTL epitopes over here that actually were reactive with this patient. And so a lot of information. And then if there is sequence from that patient, like HIV sequence, then that can be, you can link to that from down here. And that would take you into the sequence database. You could see their information. And so this is the patient detail. Let's now go on to the antibody features and that we have. So if you were to click on antibody features, you would get to a page that looks like this. A lot of the stuff that we have is downloadable. You would be able to see their sequences of the heavy and light chain of their antibody if it is available. You could see what the publication is. And you would also have something called neutralizing antibody feature. And I want to focus on that. But first, I'd like to show you an expanded view of this. And in the expanded view, you can actually see what the sequence was over here for their heavy chain. You can get to analyses using their binding studies as well. But let's go back here to look at antibody, neutralizing antibody features. And here we have, you can see there are two papers that actually are there's information from two papers over here. So let's go on to the next page and see that. Here you can see the information from the first paper where they're actually talking a lot about how there is antibody-driven selection, and you can actually see at which position they're coding that in their studies. The other paper talks about interactions and electrostatic interactions with a certain loop of the antibody with the HIV. And so there's more information about what the authors say that interaction is like. But now, so we have a lot about antibodies contacts and the features that go with it. But now let's go on to the next thing, which is the details on neutralization from the antibody. So that can also be picked from the results of the antibody search page. And
Elizabeth, just a 5-minute warning.
Okay, thank you. So here we have the neutralization data. And the neutralization data that you would get from that link that you would click. For example, if this is antibody CH235.9, we've selected, you can get IC50 or IC80 or both. But here in this example, it's just IC50. You can get a whole slew of virus names against which these neutralization details were got. And then over here you have the sequence of the virus, and you can actually scroll up and down and you can look at all of them so you can actually compare. What's really interesting and what's really good about this is that you can sort on any one of these three columns. So for example, if you were to sort on neutralization details, you could then get the best neutralizers down to the least neutralizing results of these viruses, and these would change, and you might be able to see something like, well, hey, whenever the neutralization is really good, I see that there's a methionine in position 2 or something like that. and you could begin to draw your own conclusions. Now, virus information and your alignment positions, all these things can be downloaded, and you can work offline after that. But now I want to go on more into CATNAP a little bit, because this tool is just so powerful when you're working with antibodies, and also because it links so well to this. So I'm going to go really fast now because we don't have much time. And I'm just going to go on through CATNAP really quickly. So this is the CATNAP top page that you can just get through to tools from up here. If you were to click on this, we'll see what happens. But I want to mention that you can put in custom input. If you have your own name of antibodies, you have your own IC50 or whatever numerical data, and your own viruses, you can actually put it in and use any of CATNAP's abilities. You can also do a hybrid where you put in your data, and if you're studying HIV, you can study it against, compare it to the data that is already residing in our database. But let's go on to see what would happen if you were to pick CATNAP. This is the kind of page you would get, where you could pick antibodies by names, or you could pick all of them you wanted. You could pick viruses by names, all of them if you wanted, and then you could retrieve virus or antibody details, assays, and you could also pick whether you want IC50 or IC80 and get a result. Now I want to show you another page of something like this, where you could use attributes, and I'll show you what that means, or panels of viruses. So let's look at it if you went in that way. And if you were to pick attributes, you could pick patient studies, you could pick light heavy chains, you could pick the antibody binding type and get all those antibodies into your analyses, or you could pick virus names, or you could just pick a whole virus panel, because so many panels are already set by study. And then you go in and pick what you want to retrieve and whether you want IC50 or 80 or both, and then get your results. Your result might look something like this, where you have virus names here that were picked, and here they're comparing 10E8 and PG9, two different neutralizing antibodies, and they've taken both IC50 and IC80 values. And on this side, you can see, of course, the alignment of all the viruses that you had picked, the person had picked. And then depending on what you click and how you go through, you can also see other features, like you can make a web logo of the antibody binding the epitope. And you can also analyze in other different ways. So there's a lot to be said here, but I'd like to continue on to more. And just to say, we have very, very many tools. I'm not going to run through all of them at all, but one of the new ones is GenSig, which picks out signatures and correlations. If you give in your sequences and you give in the phenotypic information that you want, then you can actually pick out certain correlations that come through. And that is automated. And so I'm just going to take you quickly through another couple tools and then hand over to Brian to finish off for us. This is a very old but very easy tool, and I wanted to show you that we don't just have complicated things, we even have things like this, where you can just, if you want to make peptides for your studies, put in your alignments of sequences and get the peptides, generate them. And the nice thing about this tool is it will kick out any duplicates that you've made, so you don't have to make too many, more than you need to, number of peptides. But here I just want to talk about Combinabber, which is the last tool I'll talk about. It's really, I'm just going to run through it. And here what you can do is you can predict antibody neutralizations through combinations of antibodies. Now, we all know that with immune therapy, usually you get much better results if you actually use a mixture of antibodies rather than just one. And so if you decide to do that, here's a way where you can theoretically see and predict what your neutralization may be, depending on the information that we have in the database or that you put in. So here would be, say, a result where this would give you the potency, and this would be the would give you breadth, coverage of how many viruses, depending on which viruses you picked, would be covered by the antibody and would be able to be neutralized. And this is a single chart, so that means each antibody singly. And you can see, for example, VRC07, this red one, actually has great coverage, neutralizing most of the viruses in there, but it's not so potent as far as how quickly it would begin to neutralize. And for example, the blue one, CAP256, actually acts much quicker. But what would happen if we were to use mixtures of antibodies? And that is the result you can see here, where you can see that if you were to use 4 different antibodies on this really dark maroon line here, you get a very potent combination, and it's also got great breadth. But on the other hand, if you were to use three of them, not 10E8, you get pretty much the same results. So really, maybe 3 would do the job. And so you can actually do a lot of predictions this way. And with that, I would like to turn back to Brian, who will give you just the briefest on our sequence database. Thank you for immuno@lanl.gov. Thank you.
Hi, I'm back and sharing the same screen. I need to go to slideshow mode. Yeah, so combination errors for 2 or more antibodies together predicts how well a combination of antibodies will do.
Sorry, Brian, it's showing your presenter's slide again.
Presenter slide again. Okay, let me stop share again. There we go. I think that should be right. The sequence database, sorry, and I'm running out of time, but we don't have many questions, so I'll run over just a little bit here and go through this quickly. We have many different tools and many tutorials you can see under our guides page. There's basic information about the database, and we have previous presentations and slideshows you can go through here. There's always help from writing to us at secinfo.lanl.gov. There's a separate email, immuno@lanl.gov, for immunology questions, but most of us read both of those lists, and we'll respond to these very quickly. The search interface allows you to search and then download the results aligned to each other. You can build a phylogenetic tree from the results and everything right from the search interface results. The search interface looks like this, where there's separate blocks of information you can search on. The top is sequence information, sequence name, subtype, virus, country that it was isolated from, year of isolation, and so on. You can search by gene region or genome region, and it's very useful usually to include fragments of some length. So if you want envelope, you might include fragments of 1200 base pairs to get things that are almost complete envelope. And then you can also search on publication and patient and so on by expanding these search boxes here. And the results look like this. And so we've searched here on Brazil and complete genomes. You can see that there's a map showing that each of these sequences is a nearly complete genome. You can sort by subtype, you can sort by country, you can sort by year, and so on. And that's often useful to sort first and then download the results. They'll come down downloaded, sorted. So you can see change over time if you've sorted on year, for example. You can also search by country this way by seeing a map of the world and clicking on any continent will take you to one search per country within that continent. But it's a good way to get an overview of what type of virus is circulating in each region of the world. We have pre-built sequence alignments, which are really handy because we take a lot of care to limit those to 1 sequence per patient. We align them very well. We codon align them so that gaps are put in between codons so they translate to protein well. Those are very useful for many things, especially for studying epitopes and so on. You'll need it aligned, and you'll need it to be able to translate to protein so that you can study the epitopes. And thanks for attending. You can contact us at those addresses. The slides are available here, and I think they'll also be available at the IEDB site. And I'll open it up to any questions now.
Excellent. Thank you so much, Brian and Elizabeth. I think we had one question come in. I'm not sure if this is for the LANL team though, if you want to have a quick look,
Yeah.
Brian.
I guess that's and it says this, will there be a standalone version of all online portals for immunogenicity prediction? And I think that's partly IDB and partly us. We do some immunogenicity prediction also. But I don't think there will be any standalone version that pulls them all together, is my guess.
No problem. Yeah, I think that I'll leave that for Bjorn and Jay to probably answer afterwards. And yeah.
Excuse me. There was one question that came in on the Q&A that I saw, and it was I believe somebody was asking whether there would be any low-level data from genomic regions annotated. I believe this is what they were asking, and my reply to that is, if it is in the literature and if it is published, yes, it will be annotated. Yeah. So, and also cryptic epitopes are annotated and their results. Thank you.
Yeah, and I also, yeah, my mic's still on. I also wanted to mention that PepGen tool is very useful. Earlier today, Bjorn mentioned writing scripts and so on to generate your peptides, but our PepGen tool can be used for any data set, any virus, and it will not only generate those overlapping peptides for one sequence, but you can put in a whole family of viruses, say, and it will generate peptides that are overlapping and tell you when you need to build more than one peptide for the same region of different genomes, because there's diversity in that region.
Excellent. Okay, I think that's all we have time for as well. There are no other questions, so thank you so much, Brian and Elizabeth, for that.